As of 2025, 2.2 billion people, or 1 in 11, lack access to clean and basic water services. Key parts of the target include providing access to safely managed drinking water, hygienic toilets, hand washing facilities, and having protected water sources that take less than 30 minutes to collect from. A prominent component of the goals surrounding water sanitation that imminently requires international cooperation include desalination of water, helping to increase water use efficiency and ensuring fresh water supplies.
Water is the medium of life. It allows ionic compounds to dissociate into ions, facilitating a range of chemical reactions and enables the transport of nutrients, waste, and chemical messages within organisms. This underpins the inherent need for clean water and sanitation practices. Microbiologically contaminated drinking water is estimated to cause approximately 505,000 diarrheal deaths each year. So the removal of contaminants like bacteria such as E. coli and viruses such as rotavirus is essential in water sanitation practices. Our target relates to molecular biology, as DNA tools such as PCR-based methods are fundamental in the detection of waterborne pathogens and allow for the monitoring of contamination in water harvesting and wastewater treatment. New research is evolving around the use of enzymes with broad substrate specificity in wastewater treatment, with the ability to catalyze the breakdown of harmful contaminants via oxidation. Desalination is another process that uses a reverse osmosis membrane method that allows seawater to be pushed through a semi-permeable membrane using high pressure. Water moves from higher solute concentration to lower solute concentration, requiring energy, hence the reverse of osmosis.
The process of reverse osmosis usually occurs in water desalination plants. Many major cities in Australia rely on these water desalination plants for a large portion of their water supply. The Sydney Desalination Plant describes 6 key steps to water desalination. Seawater is drawn in from the ocean through pipes at a very slow current to allow wildlife to swim out of the tunnel if trapped. Drum screens are used to remove any large materials such as rubbish, fragments of shells, or plant matter from the water. The pre-treatment filters remove solid materials such as algae and dirt particles from the sample. Water is pushed at a very high pressure through a number of semi-permeable membranes, which act as a filter for the salts and microorganisms, only allowing water to pass through. The water left behind is much saltier than the original sample of seawater and is then returned to the ocean. The fresh water produced is fluoridated, chlorinated, and tested before it is sent to the drinking water storage tank. The water is then distributed to homes and businesses in the region. Approximately 58% of the water used during desalination is returned to the ocean via dispersion nozzles. These ensure that the significantly saltier water is properly mixed in with the seawater to ensure uniform salinity. One of the main steps to this process is water testing. This can be done using polymerase chain reaction tests. PCR tests detect microorganisms by amplifying specific segments of its genetic material to detectable levels. This allows for identification of the organism even when it is present in minute quantities. Although this seems like a very effective way of detecting the presence of unwanted pathogens in water, limitations of this technology include the inability to differentiate between live microbial cells and dead microbial cells. So while PCR tests have potential in revolutionizing the way we test water, the technology is still being developed.
Progress towards Target 6A is assessed through tracking the Official Development Assistance, or ODA, the official financial contributions by government agencies towards promoting economic and welfare progress in developing countries. The most recent ODA report in 2022 noted an international contribution totaling $9.1 billion towards water and sanitation improvement globally. The 2025 SDG Goals Report concluded that while expansive progress has been made towards access to drinking water, sanitation, and hygiene services over the last decade, 2.2 billion people lacked safely managed drinking water, 3.4 billion without safe sanitation, and 1.7 billion lack hygiene services. From 2015, the share of the population with access to safely managed drinking water increased from 68 to 74%, safely managed sanitation from 48 to 58%, and most prominently, hygiene services grew from 66 to 80% over the last decade. Despite progress over the last decade, the goal to expand water and sanitation resources globally by 2030 is currently off track by almost 20 years. In 2020, the United Nations initiated the SDG 6 Global Acceleration Framework to increase financial contributions and thus accelerate achievement of the goal by 2030. Specifically, the initiative calls upon stakeholders such as government, society, and the private sector to help in optimizing resource funding, data and information targeting resources and progress, capacity development involving a skillful workforce, innovation of technologies, and lastly, governance collaboration. Together, this framework aims to restore Target 6A to be achieved by 2030.
With that support and effort on an international scale to improve water sanitation, we will continue to live with diseases that should have been long considered to be in the history books. In order to achieve universal and equitable access to safe and affordable drinking water for all, states need to cooperate internationally through providing technical assistance, capacity building, and financial support.